Hello everyone, my name is Zunur Iza binti Muhammad Zaidi. In this video, I'm going to share about my final year research project about isolation and characterization of chemical constituents for the leaf of Macaranga Heynei I. This project was supervised under Dr. Aisyah Saleha Kamaruzaman. Macaranga genus is from the family Euphobiaceae, comprises over 300 species, which are very rich in secondary metabolites including phenolics, flavonoids, stibinoids, and terpenoids. This genus is also known for their rich chemical diversity and bioactive compounds. Based on the previous study, Macaranga Heynei I, also known as mahang biru, belongs to the family Euphobiaceae. It can distribute in areas such as wasteland and secondary forests, and it can be also found in Malaysia, Thailand, and surrounding tropical regions. Their uses traditionally used to treat wounds, fever, and infection, and their softwood Macaranga Heynei I can be used to make paper, plywood, and particle board. In this picture show several species of Macaranga genus. For example, we had here Macaranga gigantea, Macaranga tenacius, Macaranga trilobα, and Macaranga Heynei I. And these are the plant part of Macaranga Heynei I, which is stem, plant, and scorch. Next, chemical constituents of Macaranga Heynei I that had been reported in the previous study. First, we had Malayheyneiin I, Malayheyneiin B, and Malayheyneiin C and D. And next, we also had levofolin A and B, and also we had Macarubigenosin C from the Macaranga Heynei I. Because they are rich in stibinoid and dihydrostibin compound, they exhibit strong antioxidant activity, especially in levofolin A and B, and also demonstrate strong anticholinesterase inhibitory, DPPH radical scavenging, and antimicrobial activities. Problem statement in the study: Macaranga Heynei I is reported to contain bioactive dihydrostibin such as Malayheyneiin and levofolin. However, they are very limited for the chemical study have been conducted on this species. So, further isolation and structural characterization of its chemical constituents are needed to expand the photochemical knowledge of Macaranga Heynei I and also for their genus. Alright, objective in my study are to isolate major secondary metabolites from the leaf of Macaranga Heynei I using various chromatography techniques. Second, to characterize the isolated compounds using spectroscopic methods including IR, NMR, and UVVs. So, the significance of my study to provide new photochemical data on Macaranga Heynei I and also to support further research in natural product chemistry and highlight the value of tropical plants biodiversity. Next, we go to the methodology. For the leaf of Macaranga Heynei I, we collected its total weight of 2 kg and we did the co-extraction in methanol. And then from the co-extraction methanol, we had methanol crude extract with total weight of 300 gram, and then we dissolve in methanol and water. And then from the methanol aqueous extract, we did the liquid partition into hexane crude extract, acetic crude extract, and the residue. From the hexane crude extract, which name it as MHH, with its total weight of 50 gram. And then from the 50 gram, I only took 0.97 gram to dig the column chromatography under solver system hexane and acetate. From here, we successfully isolate the compound with its total weight of 1.3 mg. So I select this fraction to further analysis using UV, IR, and MR. And then I successfully isolate and found new compound which namely as Malayheyneiin I. This picture shows the color chromatography and TLC spot and then solver system we did during this experiment. Next, we go to the result and discussion. At first, UV spectrum of this compound at 230 absorption peak at UV is indicate the presence of phenolic in the structure. Next, 260, 269, and 279 resemble the characteristic of the hydrostibin. Next, we go to the IR spectrum. Because of the small quantity, which is 1.3 mg, is very little. Because of that, we didn't get the better IR spectrum. However, these are signal appear on my IR spectrum. At here, I want to highlight the wave number at 3279, which indicate to the functional group of OH. And second, 1627 pair with the wave number 1459 indicate the C double bond C, which is aromatic. Alright, next, to elucidate the compound, to elucidate the structure of this compound, I did the 1H NMRs. This is the result from the spectrum. At the downfield region, we had type of metacarpal proton aromatic, which is at the region 6.59 and 6.43, which is at the ring B. Next, we had olefinic proton, which is aromatic at region 6.33 and 5.70. And then, we had pyrene ring at region 2.67 and 1.77, which indicate these two rings attached to the ring B, attached to the ring A. And then we had methylene at 2.78 and 2.5A, which is link ring A and the ring B, which indicate to the alpha and beta carbon here. And then lastly, we had methyl at region 1.26, which indicate this methyl group here. Alright, next carbon 30 APT NMR spectrum. At the downfield region, we had quaternary aromatic carbon, which ring at 122 to 150, and then auxiliary carbon, and then we had methyl carbon, and upfield region we had methyl carbon and also methylene. Alright, to see the correlation between proton and carbon, we did the HNBC, which is long range correlation. To see the long range correlation, this is I conclude from the 1 HNBC result. Alright, to see the interaction correlation between proton, first we look at the proton 712 with had correlation with carbon 2,6 and carbon 9 and carbon 9,14, while proton at ring 10,11 and proton at 15,16 show correlation with carbon 9 and 40, indicate that two dihydromethylpyrrole ring located on ring A, which is here. And second, the position of the dihydromethylpyrrole ring attached at ring B because of the correlation displayed by proton 7' with carbon 4', and then carbon 9' had correlation with proton 10', 11'. Alright, next, in conclusion, the photochemical study on the leaf of Macaranga Heynei I has resulted in the isolation of proposed new compound, namely Malayheyneiin I. This finding of Malayheyneiin is one of the derivative of Malayheyneiin, which contribute to the chemical profile of Macaranga Heynei I. So this study provides valuable baseline data for future natural product chemistry, pharmacological studies, and bioactive compound discovery. That's all for me. Thank you.